Is it? Okay, I think I'm ready now. I got on here and then I wasn't the host, but I'm the stinking host. Now I'm the host. All right. Welcome. Uh so happy that I got in here and the projector was working today. That was a bit chaotic Wednesday. So, any questions or concerns before we jump into chapter 2 stuff? Feeling good? Pack back good? Okay. All right. And if I start going through here, should be on the right thing. I think there were multiple ones open. This should be what's being shared. If not, and you're on Zoom, let me know if it's not changing slides. So a lot of this chapter, well, a bit of this chapter is going to kind of set the stage for some of the techniques that we think about in lab, trying to visualize organisms with the microscopes. But as I said last time, it's a lot of comparing different types of microscopes as we progress along. There are a good chunk of slides as we move through this chapter, but a lot of it really is to do some of those comparisons and introduce some of the ideas about how these microbes appear uh, through microscopy. So it really depends on the circumstances, depends on what it is that we are hoping to visualize in the first place that sets the stage for the types of microscopy that we might utilize, right? And truly a plethora of different options. The biology department has numerous specialized microscopes that potentially we get the opportunity to utilize. Just kind of depends on the situation. All right. I will tell you, uh, as a grad student, I knew we had an electron microscope. I saw it, but I never actually saw it in action. I never had the opportunity for that. My research didn't warrant needing to use electron microscopy in the first place, but I thought it looked awesome. And it wasn't until dealing with the C phages or virus hunters that I truly had the opportunity to work with the electron microscope. And my very first time, this is probably the microbiology nerd inside of me. All right. That's all fine and good, and I can deal with that. I was like a kid in the candy store. I was just like, oh my gosh, look at this thing. And I got the knobs are like, it's like Etch a Sketch, if you know what Etch a Sketch is like, the little knobs, and you can draw a picture, but it's not, unless you're real good, you're not making it's diagonal sort of stuff. Crazy, super cool. And I pulled my phone out. I'm like, you got to take a picture of me using this thing, right? And as soon as I'm finding stuff, I'm like, holy cow, and I'm taking pictures. Uh, Crazy. Uh, and as soon as I do that, I send a picture to my wife. She's got nothing to do with microbiology, right? She's looking at this picture of this virus, and she's like, that does nothing for me. I don't even know what I'm looking at. It looks kind of like staticky stuff on the old school TVs. I don't know if you even get the static, the fuzzy picture, kind of like that. And then all of a sudden, here's the hint of some sort of shape. And she's like, I don't know. I don't know what I'm seeing. It's beautiful, right? Crazy. All right. So it really kind of just depends on the potentials. There are other microscopes, some of them that the whatever you've stained will fluoresce. We'll see this in a minute as well. I was a grad student when I was using that, and it was almost the same deal. We had just a few minutes to be in there and look at our sample, and I was like, can I have a little bit longer? I'm not done, right? Uh, so some of these things I feel like are exciting, impressive. Sometimes looking at bacteria, especially the first times when you're able to truly focus in and see it under the microscope, you prepped it, you stained it, you found it, you're super stoked about it. But after you've seen a few of them through the microscope, it's kind of just run of the mill and not so impressive. But once you bump it up to some of the other technologies, I still get excited seeing some of those things. So really just kind of depends. All right. What we think about in the labs are bright field microscopy, a regular compound microscope with multiple objectives that allow us to zoom in, truly very, very magnified, but deep down, we can go a lot farther with other applications like electron microscopes. All right. So enough of that. Both of these images we're looking at truly through what we could look at through a regular microscope, bright field like this, and then with some little bit of adapting with a microscope, you can get some dark field microscopy as well. It's the opposite, right? So here light is shining through and we can see the organisms that have been stained up. And this is the opposite. It's the organisms that are actually being lit up and the rest of the field of view blacked out. All right. So enough of that. Different ways. And we've got completely different organisms up here. So this smear is some anthrax, Bacillus anthracis, boxcar shaped, rod shaped bacteria, but these are nice, kind of pretty good rectangles, sort of characteristic of anthrax. Other bacillus, typically more rounded, these pretty rectangular. Sometimes these cells will link up, forming a chain or a cord, but that's not super diagnostic with the bacilli. All right. We will talk about the cocci and some of their potentials for arrangement, and that definitely clues you into a little bit about the way we can group them. Bacillus, we might see them like this. They might be separate, not something, I wouldn't look at this, although the boxcar may clue you in a little bit, but the fact they're all linked together, not super informative. All right. This smear also has some other stuff going on in here. All throughout, we can see nucleated cells. There's boatload of white blood cells all around this slide. The majority, and we will talk about white blood cells handful of chapters down the road, all sorts of different potentials for these cells to try to combat infectious agents. And one of the first lines when we think about this could be some neutrophils. And the neutrophils all over the place in here. They're the most common type of white blood cell in your circulation. You would expect to see a good amount of them. So all of these guys with the weird looking convoluted nuclei, it is a single nucleus, but it's multi-lobed. All right. And then we've got some other cells in here, so probably some lymphocytes, B cells or T cells, kind of throughout there. All right. Next door are some corkscrew looking bacteria, some spirochetes. There's spirillum and spirochetes. And these springy looking bacteria are Borrelia burgdorferi. They're the culprits for Lyme disease. All right. And we'll touch on some more of this as we progress along. All right. So a little bit of detail about how all of this stuff works in the first place. And we're not going to get super bogged down with this. All right. We want to think about truly trying to use typically light, but sometimes electrons to focus in on whatever sort of thing we're trying to look at. And so energy, ultimately, light energy that we can see and other wavelengths of energy. And in turn, dependent upon the specifics with this, it provides the opportunity to visualize and for us to actually perceive things in the first place. So the amplitude here, we can see it's the height of the hump, all right? The wavelength of light, which is basically dictating how much energy is in there, is the space from the top of one to the next, all right? That's the wavelength. And there can be variations in amplitude and wavelength, as we see over here, spaced out farther, lower frequency, lower energy, and super packed together high frequency waves. And with that, we'll see here in a second the electromagnetic spectrum and the potential for are visible. So there's also differences in this as well. Our Petri dish, which you have worked with already in the lab, clear plastic, nice because you can look right in there without even having to lift the lid off and see bacteria that are growing on these plates. Sometimes you're lifting the lid off to deal with stuff, but you can see it right through there versus this little hunk of meteorite that is opaque. You can't see through it. And in turn, the way that light can be acting depends on the types of material in the first place. We use ground glass, magnifying, turning these things into lenses, mirrors to focus, to get the light onto the specimen and up through the scope so that we can visualize it in the first place. When we shift over to other microscopy, we might think about focusing energy like electrons and shooting those electrons like a beam of light, but a ray of energy, high energy that hits a sample. Depends on what we're talking about. And there's some other techniques we'll talk in a minute. All right. So here we got a chunk of glass, our transparent material, and shooting some light at it. We can see that light as it hits another material, there's some refraction. It bends, if you will. As it moves in here, we've got the line tracing through. So the angle changes and then it comes out the other side. Not right here, not a straight line through the reflected ray. All right. That becomes important when we start thinking about truly the mechanics of the microscope, and we're not going to dive into all of that. I don't expect you're going to be building a microscope in your basement, right? Probably not. So this, as we're looking at potentially a microscope slide, right? As the light is coming down, we can see the refracted ray as it's moving through there, the reflected, similar idea like this, all right? I think this is pretty cool, all right? So we've got a metal rod. They're probably sampling something in the water, maybe testing different chemistries, or maybe they're trying to collect some samples. I don't know. I don't know what's on the end of this guy, all right? Matters not. What we're seeing, it's not a bent rod, but it looks bent, all right? As it hits the water, really kind of making this optical illusion, and it looks as if it's bent, all right? But if you were to pull this out, it's a straight rod, all right? So it has to deal with, and this technically some physics, the refractive indices of air and water. They are different, right? And so the way that light's hitting this and what we're able to perceive looks as if it's been bent, all right? Most of the, when you're doing microscopy, most of the time, especially looking at bacteria, we will put a drop of oil on the microscope slide at our very last magnification. Drop of oil goes onto the slide. The last objective goes into that oil, and it basically acts as if the oil's kind of like a piece of glass. The oil has the same refractive index as glass. So it's acting like a continuum, and the light moves right up through it, and we don't see it refracting off, all right? Not as much, all right? So Most of the immersion oil that you use to put onto your slides has transitioned into little plastic squeeze dropper bottles that you can put a drop of oil onto the slide. Easy, convenient, all right? There are some that are still in glass vials, and you open up the lid of that, and there's a glass rod that you will pull out. It's got a droplet of oil on it, and you take it over and it dispenses it right onto the slide. Little glass rod, all right? Here's what's cool, all right? Magic, right? Optical illusion. That glass rod having the same refractive index, you can hold the vial of immersion oil up, but it doesn't really even look like there's stuff in there because it's the same. You can see the dropper at the top where it's glass and air. And as soon as it touches the oil, you don't see it at all. It's invisible, but it's there, but you don't see it, right? That I think is awesome. And I feel like we should get a handful of those in the lab, bring them back just so you can see the magic of microbiology. I think it's cool. All right. We'll show the immersion oil stuff. You're going to see it soon. All right. So different ways that glass can be ground, and we can see different things acting as lenses with the ability for light to pass through. And with that, different ways that the light could be focused, if you will, as it's moving through. Our prism, which can separate light, normal light, natural light, into the visible spectrum. You get a rainbow with a prism as the light is refracting and being separated as it moves through here. So the focal point is where we're able to visualize, and here we've got two different types of lenses. This makes me think, although these are lenses, not mirrors, it makes me think kind of funhouse mirror sort of an idea with different types, convex or concave, where images appear different. All right. So here light shining through, and we get a focal point somewhere past this as it all converges. And that's where we are able to actually visualize. All right. With the concave lens, the focal point is actually before the light is moving through there. Different. All right. Funhouse mirror-like. All right. Is the idea. So I said we'll see the electromagnetic spectrum. The visible light is a super small fraction of the entire electromagnetic spectrum. And truly within this bit, not that high of energy, all right, compared to the rest. As we move to ultra, X-ray, gamma, and cosmic, these are very close waves, higher energy, all right? Still a sufficient amount of energy as we look through this visible spectrum for life, as we think about photosynthesis and the ability for those chlorophyll pigments to get excited. There's enough energy for this, right? And in turn, we're thankful for that, all right? As we move to the opposite end, lower, the waves are much farther apart as we get down this way, all right? But this little teeny bit, this is what we can actually perceive, all right? Leaving the rest right now unable, all right? Interesting. So I mentioned the fluorescent stuff. There are potentials. There's a handful of different types of dyes that are fluorophores or fluorescent dyes that can get added, and depending upon what you're using or what you're trying to look at, depends on the dyes you may be employing. All right. So they've got opportunities to bind to different things. So here we're looking at bovine pulmonary artery endothelial cells, all right? And we've got nuclei in blue, couple, three maybe, that we can see, stained with 4,6-diamido-2-phenyl indole, or DAPI, much easier to say DAPI. The microtubules that we can see around here are in green, and this is FITSI. And then actin filaments we've got in red, all right, with the tetramethylrhodamine, or TRITSI, all right? So different structures showing up fluorescently through the microscope, and that can give you some details about specifics with the cells. When we think about bacteria, there are differences that we can utilize, and we can differentiate bacteria based off of how they're growing. We talked about before positives and negatives for the bacteria. You can see other details with them as well through some fluorescent staining, which I think is pretty impressive. So with that, we got to think a little bit about microscopy in the first place. I think this stuff is fascinating. So Leeuwenhoek, we really kind of think of him as the kind of the father of microscopy. There's a little bit, maybe others that had done some stuff as well, but what he was able to do, it was brand new, cutting edge. What he saw, people had never seen before, all right? And it's kind of mind-boggling to me, all right? He wasn't really a scientist, he wasn't a microbiologist, all right? But he was, all right? He was a merchant. He sold all sorts of different things, but he sold fabrics in the shop. And it makes me think about fabric, right? Sheets are sheets, right? But they're not, right? There are all sorts of differences with that. And dependent upon how many threads they have there, they may feel very different, right? Even though it's all cotton, right? It could feel very different, all right? And he wanted to know that what he was getting was really what he should be getting, right? He wants to look at it. Oh, it's got, you know, these threads or this many or whatever the heck it was. He would look at it and see it. And in order to do that, he wanted to magnify it. How the heck are you going to do that? I'll build it myself, all right? He built these microscopes like you see in the middle that doesn't look like a microscope to me, all right? Kind of like a little hand lens. He would grind glass, make the lens, and be able to look through that and visualize the individual threads, look at the quality of the thread, and know if he's getting what he's actually paying for, all right? Interesting, all right? So he starts doing this and starts wondering about, well, what the heck else could I look at? I want to see other things. And he starts looking at everything he could get his hands on with these scopes. And this is something that I also find a little bit mind-boggling. In the lab today, we look at something on a microscope slide, we pull that slide off, we get another one, we put it on there. We don't even think anything of it. We make them, we discard them, whatever, all right? He wanted to look at these things repeatedly, and in turn, everything that he was looking at, he built a scope for it, ground the lenses, had it, and there they were on a shelf, and he could pull it out and look at it later, all right? He built hundreds of microscopes, all right? And grinding these lenses and visualizing these things, he was making lenses and the ability to magnify and see these things like no one else. And the quality that he was able to achieve took over a century to be replicated. I think that's just crazy. And in the back of my mind, I'm like, obviously he was a time traveler. He must have been, right? It makes sense. I don't think so. All right. So he starts seeing all of these things. He starts drawing pictures. Oftentimes in science today, we don't think about drawing these things, but you could. And there are courses that are being taught in biology right now to draw scientific things, whatever it might be. And I've seen some beautiful pictures of things that students have drawn. Art and science truly should go together, all right? We used to be the College of Art and Sciences. Now we're not, all right? So different, all right? So he looked at lots of stuff. He looked at pond water, droplets of rain. He saw all sorts of things moving around, most of them probably protists, but he was able to see some bacteria. Probably not super great because his magnification was about 300 or 400 times. And to truly see bacteria pretty good, we got to ramp it up to about 1,000. All right. You could see them at those lower powers, but you see them a lot better when you kind of double it up. All right. So lots of stuff, lots of different things moving around. What the heck is it? We've never seen any of this before. All right. He calls them animalcules. He calls them wee little beasties, crazy little monsters that we don't know what the hell they are, right? Awesome. And he draws pictures, describes them, submits these images and descriptions to the Royal Society so scientists can look at them, speculate. They invite him to show stuff. He wasn't a scientist, but he ultimately became one, right? He did fascinating things, all right? Any and every sort of bodily fluid you could imagine, he looked at, all right? He's got some nasty diarrhea and it's happening for probably months and he's wondering what's going on. Collect a little, look at it. He's the first to describe giardia, all right? And it came from his own diuretic stool, all right? Awesome and gross, all right? At the same time, all right? The queen is super interested and she comes and wants to see some of these things. And he showed her many of them, maybe not his diarrhea, all right? I don't know, all right? Maybe he did, all right? Saw flagellated sperm cells swimming around. Where'd that come from? He collected his own, right? Crazy stuff. There it is up on the shelf, all right? That right there, I should just leave you with that, right? We should be done. All right. Crazy. All right. So Galileo, he was also, although we think looking at the stars, all right, he also did some microscopy and one of the pioneers with that as well. All right. So both in there. All right. Robert Hooke, this is a little bit later than Leeuwenhoek. Hooke and Leeuwenhoek, Robert Hooke, he's the one that gives us the name cells in the first place. All right. He took a chunk of bark from a tree, from an oak tree, and the type of tree is called a cork oak, and the bark is super thick, and that's where corks come from, is from the bark of those trees. So he took some cork and sliced it thin and looked at it through the microscope and was able to visualize. We got an image of, not an image, a drawing of what he was able to see, individual cell walls of the cork. And as he was looking at it, it made him think of a monastery and the little dormitories that all of the monks lived in, and they call those cellulae. And that's what he said these look like, hence the name cells. All right. Crazy. So he visualizes this. Another one, and this a little bit beforehand. Zacharias Janssen and his father Hans may have invented the telescope and a simple microscope similar to what Leeuwenhoek was built and used, all right? And then stepping it up to more of a compound type of microscope, like looking at what Hooke used just a second ago, all right? And this was in the late 1500s, very early 1600s, little bit before Leeuwenhoek, right? Right at the end, right at the start of him, all right? And this, I think, is pretty fascinating. Janssen is still a name that you can see, all right? Microscopes, other things like maybe some binoculars, they're still around. They've been around since the 1500s, right? Impressive, all right? That's a pretty good business, right? They must know what they're doing, right? And you know, if your microscope says Janssen, that's a quality scope, right? There's others as well. Most of the stuff we've got in biology are Leica microscopes. They are also high quality, all right? Super expensive. Every microscope that you guys will be working with are about $3,500. That's a lot of money for a compound microscope, all right? And the ones that have some cameras on the top, slap a couple extra $100 on there, maybe 500, because you know you want it, right? Okay, so sorta what we've got down in the micro labs, pretty similar, all right? The microscopes are binocular microscopes, so two eyepieces or oculars. The oculars have a 10-time magnification, all right? And then we move down to the objective lenses, and depending upon the types of scopes, 3 or 4 lenses on there, usually 4. You start off with a low power magnification. That one is 4x, and you multiply them together. So the 4 times 10, 40 power is your lowest magnification. And it goes up 4, 10, 40, and 100 to give you, multiplying by tens with all of those, up to 1,000 magnification. Super magnified. There are some that can go a little, bump it up a little bit, but truly the images aren't much different, all right? You don't get any better resolution or crisper image past about 1,000. Some are a little bit less than 1,000. All of our stuff goes to 1,000, all right? So there's a platform where your microscope slide will sit. We call that the stage. There's a clasp in here that grabs your slide. There are knobs down on the side of it. That's kind of the Etch-a-Sketch as well. You can move it, and it moves the slide on the stage forward, backward, side to side. And as you're imaging, as you're looking through there, you can move it around and visualize multiple things on that slide. In order to focus it, there's 2 knobs. They go together. The bigger knob is the coarse adjustment, and the smaller knob is the fine tuning. You coarse adjust on the low power, and once you find that hint of something, often it's just maybe a touch of color, now you know you're kind of in the right spot. You bump it up to the next lens and you fine tune. You don't coarse adjust again. Once you get it, sometimes you might need a little bit of coarse adjustment, but once you get it, it's fine tuning, all right? We call these microscopes parfocal microscopes. So once you get it in focus on the lower power, it's technically there on the next power up. It's just fine tuning. And you fine tune all of the way till you've got it under oil immersion. The microscopes before, like what Hooke was looking through, as we think of those compound microscopes, not parfocal. Every time you would switch to another lens, you have to start from scratch and refine it. And that truly is kind of a tedious process, all right? It's much easier, all right, with this. And I can't tell you There's no specific thing, all right? There probably is, but I don't know it. Once you find it and you've got it in pretty good focus, sometimes you've got to fine tune it away from you, sometimes back toward you. And I can't tell you one way or the other which way it just kind of depends on where you're at within that. And every time you shift it, you got to do it a little bit. And I fine tune one way, a few turns. And if I'm not really getting it, I don't keep going. I flip it the other way and I go back because it shouldn't be that far off, right? It should be pretty close. There's also times where you can't, and you go back to the other power and you do a little more tuning, try to get it a little bit better, and then you adjust it, right? It should be there, all right? One thing I will tell you, this is a heads up. When you are staining things, you're going to make your slide, you'll get it on there. We'll talk a little bit about that in a bit. But once you've done all of the staining, sometimes when that stuff happens, you drop your slide, right? Now how do you know which way was up, right? If you didn't mark your slide at all, you probably don't know, all right? And oftentimes there's a bit of stain on both sides of that slide, just depends on how you've been staining it. They're both kind of dirty, all right? If you're looking at your slide and it's upside down, so your specimen is below instead of up on the top, you almost always are able to see stuff on the lower powers. You can see a little bit. And then as soon as you flip it up and try to see it under oil, try as you might, you're not going to find it. All right. It doesn't show up because that focal point, you're not in the right spot. Your stuff's clear down here, but we're trying to focus right on where it should be. All right. So if you get to the point that you're on oil and you flat out can't find it and you dropped your slide, right? You don't know this truly is the top. You pretty much are going to know it's the top if you didn't never drop it, right? It's still going to be upright, right? But if it did, that should be something that pops into your mind. You're like, wait a minute, what if I flip it over, right? Maybe that will work, all right? Maybe not. That's where your TA comes in and they'll help you find it, all right? Okay, so there's other things. The rheostat adjusts the amount of light. When you're on the lower powers, you want to get low light. You don't want it to be super bright because you're going to be seeing a huge area. Lots of it's just light, hardly nothing stained up. And if you're staring at that for a long time, you're going to leave with a headache, right? That's not so good. Dim it down. Get it just kind of faint so you can see it. Once you start seeing stuff, by the time you get to the oil lens, you should have almost as much light going through there as you can because you're focusing in on one little bit, and the more light, the crisper the image, all right? So you do some adjusting with that dependent upon which objective you're utilizing, adjust the light, all right? There's also potentials to adjust more of that as well with the diaphragm underneath where you can adjust the amount of light that's going through. Usually we have it opened up to allow it all in there, all right? And other bits, the TA will go through some details about how to handle them, but down at the back, you can see kind of this neck piece. You're going to grab it back here, hold it by the base, take it to your bench, do the same, 2 hands, right? As you're carrying those guys. So with that, I told you you'd see the oil. This lens right here, you can kind of see it says 100 right on here. So this is the oil immersion lens, 1000 magnification with those oculars, right? So it looks like when you're going to flip it in there right above the slide, it looks like that lens is going to slam right into your slide. It gets super close to the slide, but it shouldn't actually touch it, all right? This bit of the lens right here is technically spring loaded. It will go up and down a little bit. So if you are fine focusing and you go too far, it may touch the slide, but it's not, shouldn't break your slide and it shouldn't damage the lens. But if you're touching it, you're too close. All right. it needs to be up a little bit. And you can see just the hint of oil that's kissing the top of the lens. It's just barely on there, all right? And it sucks it up and allows the light to shoot right up into the lens. Without it, you might see some stuff, but it's going to be super fuzzy, real grainy, because most of your light is refracting away and you're not getting a crisp image. If you don't put oil on that last objective lens, it's not going to look like it should. And if you put a drop of oil on there and your lens is clear up here instead of down here right by my slide, you're not seeing what you think you're seeing in the first place. All right. You're nowhere near focused. All right. It's got to be touching that oil. All right. And you should see it. All right. Unless it's upside down. After you've done that, if you're going to keep that slide, you can kind of blot the oil off. Most of our stuff goes into the box and gets discarded. All right. So just depends. This showing, I told you, you can make a bright field, dark field. You put a little adaption in there, this little stopper before the light source, and it causes the amount of light to be altered. And there you go. You get your dark field. We don't deal with dark field microscopy in lab. All right. So this isn't our Lyme disease, but more spirochete. All right. So we've got unstained samples, but we can visualize them because of the dark field. The cells show up. All right. This is Treponema pallidum. Syphilis is who we're looking at here. If you want a common name, you probably don't, right? But you've heard of syphilis, maybe not who it actually is, right? We'll touch more on that because there's some other techniques we can use with those guys to visualize. So phase contrast, a little bit different. I'm not going to go through all of these details, the anatomy of what's happening. What I think is better is to just show you what's up. All right. So this first bit is bright field microscopy, and the second one is phase contrast. Same stuff, same slide. All right. You can see the two cells. This bit up above is what I would call artifact. All right. It's probably a little blemish on the slide. All right. It does not look cellular. It's all crazy, irregular shaped, not like this, all right? These are some sort of eukaryote. I can see a nucleus, right? I can see the outline of their membrane. They look cellular, right? Looks sort of spherical. I can see stuff going on. These don't look anything like that, all right? And there's other stuff on there as well, right? Little bits in the background, right? Dust, stuff, who knows what it is, all right? It's not been stained up, so it's kind of black and white image. You can't really see details. Not like that fluorescence microscopy that showed you all sorts of different stuff going on in it, but you can still see some features, right? Then we move over to the phase contrast. It's similar, but now there's definitely some variation in here. The nucleus pops out, I feel like. So too does sort of the surface of the cell, all right? It's like, I kind of imagine it like going outside and seeing the moon, and you can look at it and kind of see craters and stuff like that. And then you bust out a telescope and look at it and really see it, and you can see some detail with it, right? From the hint of what it is to actually seeing the details with it. More like surface structural stuff, shape-wise, giving you some ideas. And our artifact glowing more, and seeing that is what makes me think it's a blemish on the slide. It looks like some kind of grooves that are in there that's making the light do that. But I don't know. All right. So this DIC is, oh, I always forget it, differential something. trying to differential interference contrast. Whew, I had to scroll down there to it because I'm like, I won't remember it. All right. You'd think I should. I always forget it. I'm like, why? Why? Because I don't deal with any of this stuff. So this image here is some fungus. We will go through some details of fungi and talk about some of the features. So this causes some infection. We're not going to spout off that right now. But you can see the cell wall, it stands out very well. You can see the reproductive structures on the fungus, the fruiting bodies or spores. And there are, I will mention it right here, differences when we think about fungi. Sometimes it's a continuum, a single kind of looks like a thread. We call those aseptate. And then there are the divisions like we have here. We call these septate. So separate cell divisions, all right? The others that look like a continuum are still separate cells, but you don't see the membrane or the cell wall in between each of them. They look different, and we can categorize based off of that. All right, that's a good stopping point. We will pick up with some immunofluorescence and other techniques Monday. Farewell, have a good weekend. Remember, pack back due today, 5 p.m.